Hello, my name is Troy McComak, and today I'm doing a presentation that Daniela Ayala and I have worked on called Carbohydrates, Function and Composition Within the Cell. Carbohydrates is one of the main four biomolecules. Another name for carbohydrates is saccharides, which means sugar, which is what carbohydrates essentially are. They are known as hydrates of carbon. These sugars can be simple or monosaccharides, meaning one sugar. They can also be polysaccharides, meaning many sugars. Carbohydrates are nutrients for the cells and give energy such as glucose. They are found in our everyday foods such as pasta, bread, and rice. Monosaccharides are considered simple sugars since it is only made up of one. It is the building blocks to create more complex sugars such as polysaccharides. To name a monosaccharide, we choose prefixes that reflect the number of carbons found in the compound, followed by what group it contains. For example, fructose would be considered a ketohexose since it contains a ketone group and 6 carbons. The formula CH2O(N) is the molecular formula N representing the number of carbon atoms. This is why monosaccharides, you always have double the hydrogen. In order to determine the group of monosaccharide, you look at the location of the carbonyl group. Every monosaccharide has one. A carbonyl group located at the end of a chain is called aldoses, while those located anywhere else are called ketose. Add in the several needed hydroxyl groups, and you have a complete monosaccharide. Each carbon has one, except for one. This lone carbon will then form double bond to satisfy the needed bonds for carbon, 4 bonds. The shapes of the monosaccharides are usually long chain structures. However, once a sugar has dissolved in a solute, it will form a ring structure called a pyranose ring. The structure allows for different things. For example, a ring structure will allow the monomer or a monosaccharide to bond with other monomers for the chance of creating polymers, polysaccharides. Continuing on with the form of carbohydrates, there are disaccharides. And disaccharides are two monosaccharides bonded together. They bond together through a process called dehydration synthesis. It is very important to know this process as we continue building upon our saccharide types. Dehydration synthesis, the process in which a hydroxide and a hydrogen atom are taken out of two monomers to form H2O or water. These allow for the carbon atoms, which now need a bond, to bond with the lone oxygen on the other monomer, thus forming a disaccharide and one water molecule. For example, look at the figure. You see that 2 glucose will bond with one loss when one loses a hydroxide and the other a hydrogen atom. Once this occurs, the two monomers bond to form maltose, and at the end you see one water molecule created as a result. Polysaccharides and oligosaccharides are a step up from disaccharides. These compounds are made of about 3 to 10 monomer bonded together through dehydration synthesis. Polysaccharides are the polymers of carbohydrates. These compounds are more than 100 of the monomers bonded together, as these monomers go through dehydration synthesis. The new bond with the leftover oxygen now becomes a glycosidic linkage. Here we can see a starch molecule, which is a polysaccharide of glucose molecules. And you can see here the starch is composed of many of these glucose molecules bonded together with that glycosidic bond here. This is pretty much how these glucose molecules can form starches. And this is why when we eat starches, they turn directly into sugar. Now here is a little activity. We will be checking for some understanding. And I want you to determine if the statements below are true or false, and then correct the false statements so they read true. Number 1, carbohydrates are joined together by peptide bonds. Number 2, a disaccharide is an example of a carbohydrate. Number 3, monosaccharides are made of polysaccharides. Number 4, polymers are made of monomers. And number 5, polysaccharides are the simplest form of carbohydrates. Here we have another activity. Based on the molecular formulas, which of the following are not carbohydrates? C3H8O3, C18H32O16, C16H32O2, C10H18O9, C4H8O2, and C6H12O6. As we stated before, carbohydrates are used for energy, but they also have many other functions. The catabolic reactions that start with digestion break down carbohydrates to use as energy, which starts in the mouth, such as the digestion of starches into simple sugars using enzymes like amylase. Carbohydrates function very uniquely within a cell in a variety of roles, like cell walls and fungi in plants. Also, there's cell-to-cell interaction is often based on carbohydrates in a lock and key type function. And these molecules are located on cell membranes in the form of oligosaccharides, which we discussed earlier. And these are called oligosaccharide ligands. In plants, carbohydrates are used as signals as well to turn on or turn off certain cellular machinery and functions. Here we have a cross-section of a phospholipid bilayer. And we can see here how there's integral proteins that have these carbohydrate groups that are attached to them. They're actually called glycoproteins, but they're integral proteins because they're embedded within the bilayer. And these glycolipids also are embedded there, and there's cholesterols and other things that create structure for that cell wall, or not the cell membrane. The cell wall is in a plant, and that gives more of a structure, which is also made of carbohydrates. But here we can see how these kind of snake-like appendages or oligosaccharides would be on the outside of the cell. And those would allow for different markers so that other cells would know how to interact with that particular cell. So here we have some oligosaccharides that are happen to have mannose, and these mannose binding protein interactions with other ligands are very unique. You can see here how the three-dimensional shapes of such interactions is very complicated, and there are different active sites on the protein that allows for a lot of different interactions to occur, and for those reactions and interactions to be very precise, so that the biochemical pathways are very solid, and the communication is very strong. Here we also have some how they would react on a bacterial cell wall, and also on the surface array. So this is a good diagram here from Taylor and Drickamer in 2009 that it kind of explains how this would how these oligosaccharides and mannose binding proteins would look. Lactose is a very interesting type of carbohydrate. It's a sugar with you can see here with the last name of the OSE there. And what it is it's a molecule of delta galactose and a molecule of D-glucose that are bonded by a beta-1,4 glycosidic linkage, which occurs right here between the two molecules. And that's what's broken down in the presence of lactase. Now I'm lactose intolerant, which means I can't digest this lactose here. And what happens is I'll eat it, it'll go right through me, it'll cause a lot of issues in my gastrointestinal tract, and it ends up not a pretty picture. But in those with a functioning lac operon, what would happen is the lactose, when it enters into the digestive state, would attach here to the repressor, and the repressor as an allosteric protein would come off the lac operon, and then it would cause RNA polymerase here, not DNA, it says RNA polymerase, to operate on the operon and result really in the creation of the proteins that make up lactase. It's been found out that there are about 3 different types of operators, but there's a lot of redundancy in that. And it's been a very well studied type of carbohydrate function for quite some time now. However, it's important to remember that in order for the sugars to be broken down and consumed in a later form, which we'll go over just in a few more slides, the sugar first has to be broken apart into more simple sugars. So this disaccharide has to separate into a monosaccharide. So after the carbohydrate gets broken down into more simple forms, such as glucose, we're going to use glucose here because we're going to be going through the glycolysis, which is the breakdown of glucose into energy, a very important function of carbohydrates. So after the enzymes break down whatever kind of sugar you're taking in, whether it be from some fructose from a soda or high fructose corn syrup, or glucose that gets broken down from your lactose when you drink your glass of milk, or other types of sugars like starches from bread. After these get broken down, and they're more simple, and we'll just look at glucose here. There's a process where glucose will go and be reacted upon until it starts to produce functional um, groups that are metabolites of glucose, and they will ultimately result in the pyruvate molecule here. So there's some energy that's given off through this glycolysis. Um, Once it starts to be broken down, there's really no going back. This is a catabolic reaction. This is an increase in entropy, and ultimately, this is how our bodies are able to uh, receive energy in the form of uh, biochemical molecules. So the importance of glycolysis is that it results in this pyruvate molecule, which is oxidized in the presence of NAD plus and coenzyme A to form acetyl coenzyme A. And this is where the tricarboxylic acid, also known as the citric acid cycle or Krebs cycle, begins. From here, the biological energy of the carbohydrates is increased due to the working of the electron transport chain in the mitochondria. So because this doesn't really happen every single time with other molecules, but when the carbohydrate gets broken down, this is the end result. And as we can see here, there's a lot of energy that's given off, mainly in the form of these NAD pluses and turning into NADH. And what this ultimately results in is ATP. And so this takes place within the cristae of the mitochondria. So it's right there, ready to be digested by the mitochondria along that electron transport chain, where lots of energy is given off. That's why they say the mitochondria is the powerhouse of the cell. Well, it's because of this tricarboxylic acid cycle that it's able to do that. And there's energy given off as NADH and FADH2s, and we can see more NADHs here. And it gives off carbon dioxide. And these carbon dioxides are coming directly from the catabolic reaction that started in glycolysis and worked its way down here to the Krebs cycle from those precursor carbohydrates. The last activity is the breakdown of complex carbohydrates like starches into simple sugars using the enzyme amylase. So amylase breaks down starches in the mouth, and in vitro the starch can be broken down into maltose through the adding of amylase. So maltose is the sugar that is created from the starches. It breaks them apart, and this amylase is present in our saliva. So that's why they say digestion starts in the mouth. Well, it's not just the chewing up the food. There's also enzymes that are being secreted in our saliva to break down food. If you've ever taken a piece of popcorn and put it in your mouth, it will be more sweet if you just kind of hold it in there and let your saliva kind of react on it. The temperature of your body will activate the chemicals within the amylase within your saliva, which will then break down those starches into maltose, and it'll taste a little malty after a while if you let it in there for quite some time. I don't really know how long you have to leave in there for it to be completely converted, maybe an hour or so, but I don't think anybody would want to hold popcorn in their mouth that long. But we can see here, we add that you can take a solution of starch, maybe it'd be cornstarch or a potato, crushed up, and you mix that with some water. And what you would do is you would put in some Benedict solution, which is primarily iodine, and iodine would then react with the starches. And what happens here is it has this, it forms this like helical molecule around um, the starch, and it forms a very colorful. It creates a dark purple color, and so it's very visible that there's starch there. Now, once the amylase works on that starch, it breaks it down, and um, the iodine is no longer able to adhere to the starch, and so the color disappears. And so here we can see an example of some of these wheat granules here that are forms of starch. Most starches are when they're broken down are very small, and they're globular here, and they turn this purplish color here. Well, what would happen is as the amylase breaks it all down, the purple would no longer be able to stay within these granules, and that can be observed through this hydrolysis test, which is kind of a good hands-on experiment to show to students. And oftentimes it's having to be put into a beaker and stirred up and heated up, and you just spit into the beaker, and eventually it turns from a dark purple to clear, and or like a whitish color. And it's a very interesting experiment that a lot of students um, get a lot out of, and it's kind of gross, but it's kind of fun too. So yeah, this is a pretty fun activity. Well, I thank you so much for your time and listening to this presentation. I really appreciate all the effort that my partner put into this presentation, and I hope you have gained some insight from it and that you're able to teach this information to your students. Here are some references that we used in the presentation, and here are some more references. Thank you so much and have a wonderful day. God bless.